So what we're going to talk about now is T cells, so-called cellular adaptive responses. So cellular responses, unlike humoral, mediated specifically by cells, not by antibodies, by cells. Cells are effectors. Now let's talk about the evolution of T cells, what happens in case of T cells. So T cells originate like any proper white blood cell in a bone marrow. And then they mature, but this time they mature not in the bone marrow, but in thymus. So what happens in thymus? Stays in thymus. Okay, two things that happen in thymus are negative and positive selection. So what is positive selection? Ability of cells to recognize so-called major histocompatibility complex molecules. Okay? Now, is this a yes-no question that I ask here? Do cells recognize MHC? Is that yes-no? By the structure, yeah. So if the answer is yes, they do recognize MHC, then cells go to negative selection. If the answer is no, then these cells go to apoptosis. In negative selection, the question that is being asked is, do cells attack self-antigens? And the answer is, if answer is yes, then they go to apoptosis. If the answer is no, then cells go into the lymphoid organs. So far we good? Now let's pause for a second and let's talk about the enigmatic MHC. So MHC stands for major histocompatibility complex. Now, let's break down what it means. Look at the board. Complex refers to the fact that it's a bunch of proteins, okay? Good? Major, most important. Histocompatibility. Histo means tissues, right? So compatibility of tissues. Essentially, MHC proteins allow cells to distinguish between self and non-self. Let me give you an example. So imagine that my arm, look, my hand, is MHC molecules, okay? Do you see this? And they stick out some antigen on the surface of the cell. Look at this blue antigen. What does that mean? For your immune system, this blue signal means this cell, it's fine. This is self. I'm self. You don't need to kill me. I belong to that body. Okay? Now, cell gets infected. And on its surface, MHC, molecules MHC, starts to show, not the blue one, but the red antigen from whatever infects the cell. Does that make sense? Now, your immune system sees that. And what does that mean for the immune system? It means that this cell isn't normal anymore. It has to be destroyed. Does that make sense? We clear? Now, good example. Let's say my marker is blue, and for Michaela, the normal marker, normal, is green. So if we take my kidney and put it in Michaela's body, for me, my kidney is fine, right? But for Michaela, whose immune system recognizes green as normal, Michaela's immune system will see blue kidney and will freak out and will think that something is wrong. Does that make sense? This is why we have the problem of rejection that we will get back to. Does that make sense?
But even if we have the same blood type?
Absolutely, even if we have the same blood type.
We can have different colors.
Of course. The closer, I mean, this is why we'll look at, basically this is what physicians are looking at when they find a donor, a matched donor. MHC is the general term that applies to all mammals, basically. Are you following me? In humans specifically, this group of proteins is called HLA. You don't need to memorize it. Human lymphocytic antigen. Okay? Now, basically, there are standard techniques to look at similarities between HLA of a donor and HLA of the recipient, and the closer the match, the better. You can determine this based on the blood. So when, let's say, I am willing, like, okay, I'm dead. My organs are going to somebody else. They will take somebody else's blood, they will take my blood and identify my HLAs, and they will find a donor that my organs are suited for. Does that make sense? So it's not like, okay, here we have a donor, well, let's see from which, basically, well, kind of, yeah, but when a person dies in a car crash and the person is an organ donor, they will find the best match. It may not be at the top of the list, at the very top, because maybe, whoever's on the top, it's not really a good match. We're going to lose the organ and we're going to lose the person. It's going to be just a waste. So they have to balance the urgency and the match. Clear? Let's move on to this. So there are two types of MHC. Type 1 MHC on all nucleated cells. Now type 2 is only on antigen-presenting cells. Or so-called APCs, which include dendritic cells, macrophages, and B cells. Okay, we good so far? Now I'd love to go into kind of explanation what's the deep mechanical difference between type 1 and type 2. Unfortunately, I think time constraints don't let us do that. Here's what I want to tell you something. Listen up. One of my favorite questions. It's a super easy question, which trips people like crazy. It's really easy. It's 0 out of 10. So well, first of all, before I ask that question, what are non-nucleated cells in a human body? Red blood cells, right? Platelets and sperm cells. So basically, obviously only in males, sperm cells. Okay, we're good? So 3 types. So I'm going to ask you a very simple question. Which MHC type do antigen-presenting cells have? It's an open answer, open-ended question. It's an easy question.
Which type of, can you repeat the question?
Yeah, which type of MHC molecules do antigen-presenting cells have? Do they have type 2? Do they have type 2? Yeah. Do they have type 1? Yeah, they nucleated, have both. Antigen presenting cells have both type 1 and type 2. So I'm saying that type 2 can be found only on antigen presenting cells. Does it make sense? One more question. What do antigen presenting cells do? It's another simple, stupid question. Look at the name. What do antigen presenting cells do? Huh?
Is it antigen?
Presenting cells. Antigen presenting cells. What do they do? Present what? Antigen. That's it. They present antigen. To what? The answer is to T cells. Okay? So T cells, I call them dumb cells. Look, like B cell, remember we were talking about B cell just a little bit before. B cell technically can recognize antigen by itself. T cells are stupid. They have to have an antigen presented to them. Am I clear? So, look what happens. Let's get back to where we left off with the T cell responses here. Check this out. It all started in the bone marrow, and they went through maturation in thymus. So now you understand what positive selection means. It means that T cells are tested for their ability to recognize MHC, for their ability to find these cells, the bad ones, and to destroy them, right? Because they need to see this whole thing, okay? Does that make sense? So that's positive selection, to recognize MHC. If positive selection fails, think what happens. You're going to have a whole bunch of T cells. Some of them are going to be functional, and many of them are not. Because if they can't recognize MHC, they are useless. They're still T cells, just useless. And then negative selection. Now that you eliminated all useless T cells, you need to eliminate all zealous ones. You need to eliminate T cells that can attack your own tissues. Okay? So if they can attack self-antigens, they should be eliminated. What happens if negative selection fails? You get an autoimmune disorder. You see what I'm saying? You get a population of T cells that will attack your own tissues. Clear? So, now we got all the selections done. They are in the lymphoid organs. What happens there? There happens an antigen presentation. Usually, check this out, usually, most cases, antigen presentation in the lymphoid organs occurs via dendritic cells or macrophages. Okay, we good? Mostly dendritic cells. Now, what is antigen presentation? So, look at me. I'm going to play a role of the dendritic cell. And as a dendritic cell, I have both type 1 and type 2 MHC, right? So what I do, look, type 1, type 2. I stick it out, the antigens, and I tell T cells, look, you see this? You see this pink stuff? This is what you need to kill. This is what you need to deal with. This is the pathogen. This is the enemy. Go and find pink and help to destroy it. Is that clear? Now, why two ARBs? Why two MHCs? Turns out there are two big populations of T cells, big types. So if antigen is being presented via type 1, MHC, look, antigen is presenting via type 1. It activates CD8 T cells. If antigen is being presented via type 2, MHC, it activates CD4 cells. Now, first of all, couple of my favorite bits here. Generally speaking, the fact that CD8 T cells only recognize type 1, while CD4 cells only recognize type 2, this is called MHC restriction. Does that make sense? Are we clear? So again, type 1 recognizes CD4, or the other way around, sorry, CD4 T cells are presented via type 1, CD4 type 2, CD8 type 1. What's on the board is correct, sorry, I misspoke. I usually call this rule always 8 rule, because 8 multiplied by 1 gives you 8. 4 multiplied by 2 also gives you 8. Does that make sense? Tongue-in-cheek question. Are there exceptions? Yeah. I learned about them 3 months ago. I mean, you don't care about those. There was a paper, well, not 3 months, about a year ago. There was a paper showing that in some exquisite cases we can observe blah 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 violation of MHC restriction. But for our intents and purposes, listen, for our intents and purposes, this is fine. Okay? MHC restriction is fine. We're good so far, right? Now, what is the difference between CD8 and CD4 T cells? So CD8 T cells, after they presented with an antigen, they differentiate and proliferate, just like B cells, into memory CD8 and effector cells, which are cytotoxic lymphocytes. Or CTLs. Look at the name, cytotoxic. What do you think they do? What do you think they cause? Cytotoxicity. We're gonna talk about the mechanism in a second. What about CD4s? CD4s develop into, well, memory CD4 T cells, and then they develop into T regulatory, okay, and T helper cells. So this is, for me, the hardest part of explaining the function. I can tell you why. Because I have to talk about a few things that happen at the same time, basically, like they aren't. Okay? So, absolutely kind of randomly, I'm going to start with this T helpers. What do they do, based on the name? They help. They help. And they help in 3 major ways. Check this out. They first can increase B cell activation. Am I clear? So they can improve. Remember I told you when we were talking about B cells, how they can recognize antigen and make antibodies, but like meh. So this is why they really need T helper assistance. You see, if T helpers are engaged, B cells will make antibodies much more efficiently. Is that clear? Does that make sense to you? Then they increase macrophage activation. So in some way, look, I'm going to play different roles now, various roles. Look at me. Like I'm dendritic cell. I hold the antigen and I tell CD4 cell, hey, fella, see this? This is the enemy. So CD4 cell runs to macrophage and says, dude, I saw this recently, let's go, and you will kill it. Not just you will like destroy everything. I will draw you to the side where the pink ones are, and you're gonna like clear the side. Okay? So it makes macrophages specifically targeted. Like CD4 T cells will draw them to the site of infection, so to say. Am I clear? And finally, T helpers help the development of the cytotoxic lymphocytes. We good? Clear? Clear.
What's that like and then next to that?
It's macrophage. It's a short for macrophage. It's a pretty common way to name it. Does that make sense? Now, what is the function? How do CTLs work? So here's your CTL. And here's an infected cell. You can kill me. I don't remember which one is produced on what. Basically, there's a receptor and a ligand. They're called fast receptor and fast ligand. Let's say it's a receptor. I don't remember for sure. It's a receptor. Here's the ligand. See that contraction? So like the CDL and the T cell will kind of shake hands, okay? And after shaking hands, what's going to happen here? Apoptosis is going to happen. Okay? Another option is that your T cell here, cytotoxic lymphocyte, it will produce so-called perforins that will make a perforation in the pathogenic cell, and then it will release so-called granzymes. And granzymes will float into the infected cell and also result in apoptosis. Okay? Are you with me? So, again, what happens, one way or another, either through ligand receptor interaction or through the release of perforins and granzymes, cytotoxic lymphocyte induces apoptosis of the infected cell, or malignant cell for that matter. Does that make sense? Now, I have a question and I ask you honestly. Does that sound like really, maybe not boring, but really fundamental? It's basic immunology. Now, let's run some clinical stuff here. First of all, look at this scheme and tell, oh, we didn't talk about Tregs. I'm just going to say Tregs, they downplay your immune responses. They kind of reduce, they suppress it. Because you can imagine that immune response that lasts too long is not a great thing, right? Because it can damage other tissues. Make sense? Cool? Okay. Now, here's some first question that I want to ask you. What is the worst cell type among all T's to lose? So they won't be just killing, right?
Wait, what?
What is the type of T cells that you definitely don't want to lose? That will have the most profound, losing which will have the most profound effect on your immune system.
The helpers.
Your helpers. Because think about it. If your helpers are gone, what's going to happen? Your macrophages are going to suck, right? Your B cell responses are going to suck. and your CD8 responses are going to suck. See that? This is what happens in HIV. HIV specifically infects CD4 T cells. And Tregs also, people suffer from autoimmune disorders. So if you let HIV run rampant and CD4 cells are gone, the entire immune system just crashes completely. Does that make sense? Are we clear? Another clinical application. What if you're losing Tregs? Specifically, or Tregs are dysfunctional. Remember, what do? They suppress the immune response, right? If you don't have a suppression of the immune response and your immune response runs rampant, what will it result in? The rampant immune response. Your immune system will start attacking your own tissues. Does that make sense? We call it an autoimmune disorder. It's been shown in many people with autoimmune disorders that Treg function is altered. In people with IBD, inflammatory bowel disease like Crohn's colitis, Tregs are all messed up. Is that clear? Now, here's a cool thing. So far, from what I presented you, I hope you got an impression that our immune system is extremely robust. Right? It's versatile, it's robust. So let's say I was a normal cell. That's my MHC type 1, right? And I turned into a malignant one. I'm a cancer now. Cancer is different. I'm not blue anymore. I'm different, right? Immunologically different. So in an ideal case scenario, your immune system will recognize this, right? And your CD8 T cells will run, see that, kill me, induce apoptosis. And I want to highlight only apoptosis, no phagocytic activity, nothing like this. It will kill me. It doesn't happen, obviously, because humans have cancers. Why? I have so-called checkpoint inhibitors. In a very, very poor and simple analogy, what I do, I have a capacity. Let's say you are T cells and you want to kill me, and I have a capacity to tell you, chill out. I'm just inhibiting you. Chill out. And just, oh yeah, screw that. We're going to go have a drink, Like T cells in cancer patients. Some cancers can produce certain molecules by interacting with, when they interact with T cells are losing the drive to kill. You see what I'm saying? So a couple of scientists, one from MD Anderson down in Houston and one Japanese fellow from, I think from Tokyo, they independently developed a phenomenal drugs called the checkpoint inhibitors. So what they do, in essence, look, let's say I'm cancer and this is my checkpoint thing. So this is how I inhibit you, okay? So checkpoint inhibitor drug will take this capacity away from me. And T cells are like, holy crap, we didn't notice that. Let's get rid of it, okay? Checkpoint inhibitors are extremely expensive now, but have you seen the ads? Opdivo and Keytruda. Two drugs, I don't remember who makes them. One is Bristol Myers Squibb, I don't remember who's the second maker. They are pretty successfully used in treatment of non-small cell lung cancer. Not that they cure, but they very much extend the life expectancy in patients with lung cancer. Did you hear about Jimmy Carter having melanoma a couple of years ago? He got treated with checkpoint inhibitors and he's in complete remission. They don't work against everything, but they work against certain types of cancers super, super well. And it's something they got Nobel Prize for, those two fellows. Okay? Just showing you that, there are different interactions here. And this is why, like, mainly, all attempts to cure cancer, well, most attempts to cure cancer, are focused on creating some kind of a vaccine to stimulate T cells to destroy tumors.
So in cancer, the T cells not destroyed because something else tells them to like.
Cancer tells them to chill out. I can tell you more. It's kind of funny. I love the name. Macrophages can also destroy malignant cells, technically. So T helpers can activate macrophages to destroy malignant cells. Turns out cancer cells produce special signals that are called, I'm not making this up, don't kill me signal. It's called don't kill me signal. It's targeted at macrophages, and they tell macrophages don't kill me, and macrophages don't kill them. And if you can inhibit don't kill me, then they start working. It's super funny. Okay? So the attempt, one of the attempts is to activate a T cell arm of the immune system so that it can eliminate cancers. Clear? Let's take a break.